Thank you very much, Sarah, for agreeing to give a Build a Cell seminar, and you can get started.
Thanks, Kate. I'm really excited to tell you about some of the work that I've been doing in my lab. This work is actually new in the past 2 years. It's supported by a National Science Foundation grant from the program Molecular Foundations of Biotechnology. So I am a researcher at a primarily undergraduate institution, Central Connecticut State University, and I'm collaborating with Fordham University, which is also an undergraduate university, and we are working to build membranes for different applications. And my research is astrobiology, and so in this example, we're focusing on origins of life, but you could imagine formulating membranes for other synthetic biology applications as well. My background actually kind of is a synthetic bio application, which is during my PhD, I was trying to build a photocatalytic protocell that would make precursor molecules into membrane forming material using a Rubipy catalyst. And the idea is that if we could couple that and the production of information, that we would have a usable protocell. So very synthetic, non-origins of life beginnings to my research. Yeah, so let me tell you, I'm going to start off talking a little bit about lipid membranes. I'm not sure what the expertise of the group is, and I'm a biochemist. I really am a colloid chemist, but I teach biochemistry at my current institution. And so I'm going to just do an overview of what lipid membranes look like. So when we talk about membrane forming lipids from the origins of, or for modern biology, not for the origins of life, we think of it as having a head group, which is here as this little red circle. And in biology, there's a large variety of different head groups. So a lot of them have phosphate in them, so they're negatively charged. The simplest being phosphatic acid, which just has a proton in the molecule. But we also have these bigger head groups like amine-containing head groups, or even like the phosphatidylserine is zwitterionic, or things like inositol or sugar, right? If we have like a glycerol there. So head groups are pretty diverse in biological membranes. And it depends on what specific cell membrane type you're looking at. So there's a lot of difference between mitochondrial membranes, plasma membranes, cell membranes of bacteria. There's a large amount of diversity that's not even represented on this page. Well, also, when we think about the tail groups for bacterial and eukaryotic lipids, these tail groups, so the black part here, have a huge diversity of different options. The way that we build fatty acids is through acetyl-CoA additions. And so these are all even numbers of carbons. And then there are some places where there's unsaturation, and these double bonds create a little bit more membrane fluidity. So an organism might change its fatty acid composition and its head group composition based on the conditions it's growing in, or in organisms based on the cell type that it has. So I hope that I have impressed upon you that membranes have lipids for membranes are very diverse class of molecule. They also are non-symmetrical, so the outer leaflet and the inner leaflet have different compositions of membranes, or of lipids for forming membranes. So the organism is regulating what lipids are in the membranes to determine function, presumably. Okay, it even gets more diverse because we have one other lipid type, which is made of these isoprene groups, which are 5 carbon groups instead of 2 carbon groups. And you can see these little black dots represent where you would cut those off as 5 carbon units. And so the isoprene lipids tend to be 5, 10, 15, 20, not even numbers of carbons, and the molecule folds up into this very interesting structure, which then can cyclize. And I don't know if you recognize this kind of structure. These isoprenoids are the structure that we use to make cholesterol. And we all know that cholesterol also modulates lipid membranes, right? It changes the membrane fluidity. And so you might need to add some isoprenes in. But these isoprene lipids can also be used instead of fatty acids to make our hydrocarbon tails. Now, bacteria don't do this, right? Bacteria use fatty acids like are shown here, but in archaeal membranes, they use these isoprene lipids to generate their hydrophobic tails. Archaeal lipids are also different in two other ways. I don't know if you can tell from the way that the glycerol backbone is attached to our hydrophobic tail, but they are the opposite chirality. So this is on the phosphate head group is on the third carbon here versus the first carbon here. And then they also are different because this bond here is an ester bond, whereas in archaeal lipids, we have an ether bond. And so the ether bond is considered more stable, and that means that allows archaea to live in more extreme conditions, perhaps. Archaea also sometimes use these tetraether lipids, which are, they have two head groups. So these two head group amphiphiles actually span the membrane. So they go all the way across the membrane to make it's a monolayer instead of a bilayer, right? So a bilayer is like two sets of molecules, but you can actually get these tetraether lipids to span the membrane of these archaeal membranes. So there's not only within like bacterial lipids or eukaryotic lipids, but also then in a totally separate branch of life, there's also a huge diversity of lipids that are used to form membranes. In none of our biological systems do we have a single lipid that is composing all of the membranes in that cell. We always have a mixture of different lipids to make our cell membranes. Yeah. And so I guess that is an observation that likely if we are going to make a synthetic biological membrane, we want a mixture of lipids for that, including for the origins of life. And there's something interesting about there being such a big divide between bacteria and archaea in thinking about an evolutionary process, which kind of indicates that a biological system that needs a membrane is going to use whatever is most convenient, probably metabolically for it, but that the membrane itself has a function. And as long as it's performing that function, the exact identity of those lipid-forming molecules doesn't matter so much. So it's going to use whatever molecules it has the best opportunity to use to make those membranes. I think there's also something really interesting about the divide here that I don't have time to talk about, which is, you know, DNA and RNA are very, very similar. There's not a lot of huge diversity there. We only have one example of how you make information, and that's through these base pairing opportunities. Proteins are very, there's not a lot of diversity in proteins, right? It's, 20 about alphabet, and they're all alpha amino acids. Yeah, so I think having such different lipids between archaea and bacteria is a really interesting opportunity for understanding what like alien life might look like, because they are very, very different, but they share some of the same rules. All right, so back to thinking about origins of life. If we are going to make membranes 4 billion years ago, what would we use, and how do we do that? So this is a little bit more review. So the way that we think hydrophobic molecules were made for in the absence of biological metabolism is through a Fischer-Tropsch type synthesis. And in this summary from 2018, you can see that the products that were found from this type of synthesis, which happens when carbon monoxide interacts with hydrogen gas at high temperature and pressure, presumably in the crust of the earth in the presence of a metal catalyst, and we produce these hydrocarbons. And so in the synthesis that was done in the lab, the most abundant hydrocarbon was a normal alcohol. So in the case here, this would be like heptanol with the OH group on the end. And you can see that there's actually a lot of different carbon lengths in this molecule, up to 38 carbons long, so pretty long. And they have a bit of a Gaussian distribution, so it centers on maybe like 15 to 20 carbons. The next most abundant hydrocarbon is an alkane, straight chain alkanes. And if you're thinking about origins of life membranes, then you probably want decanoic acid, which is in the alkanoic acids category, and that is around 13% of the molecules found. You can see that there's some other molecules like alkyl formates and other alcohols that are present from these reactions. So it's not, when we're making amphiphiles, there's not a good synthesis that will make a single amphiphile of a pure compound in an early Earth environment, which is one of the challenges of doing origins of life research. We have a lot of byproducts because they're not made with enzymes. We've known for a very long time, so this was published in 1978 by my PhD advisor, David Deemer, that if you mix fatty acids, long chain alcohols, decyl sulfate, and you adjust the pH in water, that you get membranes to form, vesicles form, that they have a very specific pH need, that fatty acids are sensitive to calcium fluoride. All of this was published in this first paper. And since then, we've added more and more different amphiphiles in through new discoveries. So these glycerol monoalkyl esters, long chain phosphoesters, amino acid esters. So, more and more head groups for our decanol, essentially, or decanoic acid. Recently, in fact, this year, Ram Krishnamurthy's group published this idea of a evolution of membranes. So the origins of life started with something fairly simple, like shown here, decanoic acid. You can actually add a glycerol to that just through dehydration in the presence of glycerol and DAP. This is an activated phosphate group, and that gives you this glycerol monodecanoate, GMD. And like this is better at forming membranes than our first molecule. And then, you can add a phosphate to this, and this is even better than the previous two molecules. And so there's like this accretion of better and better membrane formers for the origins of life. And this is kind of the grand hypothesis that eventually we somehow get to these perfect phospholipids for making cell membranes. So the question that I'm trying to answer is, how do you get decanoic acid or a single chain amphiphile to form a stable membrane at the origin of life if it is really very simple, if it's a really very simple vesicle? And what compositions, if we are working with mixtures, are going to allow membranes to form? And also the null hypothesis, what compositions do not allow vesicles to form, or in what compositions do we not see vesicles? So we have proposed that we can use a lab automation and computer modeling to help us better understand these compositions. And the compositions the automation that we're using is that we have a liquid handling robot that is working in 96 well plates or 384 well plates. to generate all of our different compositions. We are using a confocal microscope with a motorized stage to image all of our wells in our 384 well plate. Then all of those micrographs are processed using it's a template matching computer algorithm. And we are then working to understand the compositions using some machine learning techniques. And then using Bayesian optimization, we are predicting certainty of whether or not vesicles are forming in a specific composition or not, and designing our new experiments from that. So this is a iterative cycle. We've just finished round 5 and we're in our final round. So this is like in preparation for publication. So our amphiphiles that we've started with are fatty acid, and here we're using the 10 carbon amphiphiles. We also have the 8 carbon amphiphiles, but we haven't had time to test those yet. And we are counting decanoic acid, the protonated form of the fatty acid, as separate from decanoate, the deprotonated form. So those are this one molecule is kind of like 2 of our different amphiphiles. We have decanol, decylamine, trimethyl so primary versus quaternary. The GMD that I was talking about in that last slide, and then decyl sulfate. The reason that we wanted to include these amines is because all life uses positively charged amphiphiles. In fact, the phosphocholine and phosphoethanol amine are very, very popular head groups for biological membranes, and there's very little research that has actually been done on how that composition impacts vesicle formation. We also are using decyl sulfate as another negatively charged molecule because the decyl phosphate is actually pretty, it hydrolyzes pretty quickly. And so instead of using the decyl phosphate, we're using the alkyl sulfate. Also, decyl phosphate is not commercially available. And okay, so we take these 7 amphiphiles and we are mixing them in we had our computer program generate 10 million different compositions, and we randomly chose 48 of those. And in our first round, we limited our total concentration to 20 millimolar. So we said, hey, we don't want any compositions more than 20 millimolar. So you can see that in composition number 9, we have a little bit of decanoic acid and decanoate. We have a whole bunch of decyl sulfate, and then a little bit of decanol and glycerol monodecanoate. And yeah, okay. And then the total concentration is about 7.5 millimolar. And okay, so let's talk about what happened. We made this composition and we rehydrated it in water, and then we checked it out on the microscope. And when we did our automated micrograph analysis, it's using multi-scale template matching. So we essentially build a template. Here we have 3 different templates that look kind of like this. And it says, oh, how close do the things in the micrograph look like our template? And it counts them. So in this micrograph, there were 29 vesicles found. All right. So in our first round, every single sample had some vesicles detected using our micrograph analysis, except for sample 17. It was the only one that didn't have some vesicles. We're doing them in quadruplicate because there's a little bit of variation in how you rehydrate the samples to how many vesicles you find. And so we found that having more than duplicate was important. But yeah, there was one sample in round 1. So I think we did very well, but it's problematic if you're trying to learn where vesicles form and they form everywhere. And so we decided to reduce our total concentrations. And so in round two, we had far fewer samples with vesicles. So we had lots of negatives, but also we had some samples that had many, many vesicles found. And now that we've been doing this for a little while, we have had some samples that have had like close to 1,000 vesicles in 4 micrographs. So like very, very high concentrations of vesicles found or liposomes found. And here we're selecting for our next round of compositions, we're selecting
There it is.
For round 3. We were selecting for where we weren't sure whether vesicles were formed or not. So we essentially calculate the entropy and we then choose the 48 most uncertain samples so that we can, like if the program thinks it knows that there are no vesicles, it doesn't choose those samples. If it thinks there are lots of vesicles, it doesn't rerun those samples. It always is picking the ones that we're least certain about. We can try to understand our samples using principal component analysis because it's hard to imagine things in 7 dimensions. I will say that there's not a relationship. So from this, there's not a relationship between the total amphiphile concentration and the number of vesicles found. It looks like a grid, not a line. We can use principal component analysis to try to separate out vesicle formers from non-vesicle formers. And you can kind of see here that there's a bit of separation, although it is not very clean in just 2 dimensions. There's quite a bit of overlap between vesicle formers and non-vesicle formers, although they separate out somewhat in these 2 components. And it looks like from this that our charged amphiphiles, so decanoate, decylamine, decyl trimethylamine, are generating a lot of the variation in PC1, and that decanoic acid is generating a lot of the variation in PC2. That's our principal component analysis observations. I think it's hard to draw more information from it just because this is so poorly resolved. Okay, so the other observations that we have from this, from these results so far is that when we put in too much glycerol monoester, we have very, very little vesicle formation. So pretty much as soon as the GMD concentration gets above 10%, we have almost no vesicles in our samples. So that I think that's like a really interesting, if GMD is like one of the evolutionary steps in making vesicles, then how do you ensure that it is not getting to a place where the vesicles lose fitness because there's too much around before getting to that next step in evolution, right? The phosphoglycerol. One of the other really interesting observations was that when we looked at turbidity, and I thought, because I've been doing this for a while, I was like, oh, if the sample's turbid, then it probably has vesicles. And it turns out that a lot of our samples that have almost no turbidity have vesicles in them. And so we actually couldn't use a quick, I was hoping we could like quickly like scan a plate and it would be able to predict whether or not a cell had vesicles so that we wouldn't have to do microscopy on it, right? Like if it's not turbid, then don't do microscopy. And it turns out that a lot of our very low turbidity solutions have vesicles present. And then the last thing is that we have now made bigger volumes of these samples and determined the CVC of some of our better performers. And we do have some mixtures that have sub-millimolar range vesicles. And so we can actually get our amphiphile compositions to a place where they'll form membranes at less than 1 millimolar of total amphiphile, right? So you actually need very little material in solution to get vesicles to form. All right. The other thing that has happened from this project is that we have a lot of questions. This is a great opportunity for exploring different compositions of liposomes. And so we are thinking about this from a lot of different angles. So moving forward, trying to answer the question, how do we get functional vesicles, and thinking about what that means. What is a functional vesicle? And also thinking about this from a biotechnology point of view. If you have an application, like you're trying to build a synthetic cell and your liposomes seem to not be functional, can we do a compositional formulation experiment to help optimize the function for those synthetic cells? And maybe thinking also a little bit about biotech and pharmaceutical applications. All right. I'd just love to tell you more about the team of people that are working on this project. So my postdoc is Christelle Icoso here in red, and she is a physicist who has been doing all of the coding for lab automation and thinking about like detecting pH using indicator, and how do we automate pH detection. Avery Glagovich is working a lot on our functional membranes, and he's also been working a little bit with getting us some archaeal liposomes to do some tests on. Autumn Sankofa is working on some of the environmental constraints. So what are the best liposomes for a variety of environments? And then in the center here, Joshua Schreier is our computational chemist, and his postdoc, Hao Lu, is a materials scientist who is doing a lot of the Gaussian processing optimization and our machine learning side of things. And we have 2 summer students who are working with Avery doing an assay, a calorimetric assay. The one of the really interesting things that has come out of this is, if you can see this vesicle here below, this is from our confocal. This vesicle is like 95 microns in diameter. And so we have some, it's like, so if you could see fluorescence really easily, you might be able to see this with the naked eye, which is really, really exciting. And yeah, we have some really, really large structures present in this. So thanks for coming in on a Monday and listening to me talk about liposomes. And I'd love to answer any questions you have.
Thank you so much. I really like the talk, but I love imaging in talks. There's a lot of questions in chat. Can you see chat?
I can see chat.
If you can read the question out loud, because people watching the Zoom recording won't be able to see the chat, so read it first.
Yeah. So it says, why does decanoate get split? And the other lipids in the protonated states don't get separated. Does the fatty acid charge make much more difference than the charge on other head groups? So the answer to that is where the pKa lies. So the quaternary amine is always protonated. The pKa of decyl sulfate is somewhere around like -2. And so we just don't expect environments to ever protonate the sulfate group. But the decyl amine, so the primary amine, I think I can go back here. The primary amine has a pKa of about 9.3 or 9.4. So you can actually, in the samples that we have here, the primary amine is actually protonated. And if we took the pH up to 10, then we would see a deprotonated form. But I think we were trying to work in a more neutral range of pHs. So the protonation state for the fatty acid going to the deprotonated state is about 7. And that means that at a physiological pH, we're likely to have both present in solution. Yeah, I think I have done experiments where we go up to pH 10, and so the alkyl amine exists in both protonation states. And it performs a lot like the fatty acid then, because it's polar and not charged. Okay, so the next question is, did you consider possible order of assembly? For example, making fatty acid vesicles first and then adding esters. Something about mimicking the evolutionary pathways of increasing complexity. I have not, but other researchers have looked at starting with a single membrane group and like slowly adding in micelles that then get absorbed by those liposomes. And you can make vesicles grow that way. In our work, we really wanted to start with complex mixtures. Oh, sorry, I didn't mean to use the word complex, with diverse chemical mixtures to understand how in an early earth environment where you're not purifying molecules, how those form into membranes. And I think if you make one type of vesicle first and then add in other things, you may bias the structure. But maybe if with enough time, heat, and cycling, it would just all come to the same solution anyway, right? Like on evolutionary timescales or geological timescales, what we're putting in first probably doesn't matter. Okay, so the next question is, could you elaborate a little on the workflow? Were vesicles, liposomes formed in just one rehydration cycle, no extrusion? With the image analysis, were you looking for vesicles of a certain size? So the way that that's a really good question. The way that the robot operates, it takes our amphiphiles that are dissolved in an ethanol solution. We tried a bunch of different solvents for this: ethanol, ethanol water, isopropanol, and all of them dissolve in ethanol except for the deprotonated form of the fatty acid and the decyl sulfate. So those are in a water solution. And then we dry it out. So we make the plate in ethanol and then we remove all of the liquid and then we rehydrate with water. So it's a pretty good mixture of solubilized amphiphiles that are dehydrated. And then once they are rehydrated using the robot, we pipette up and down to mix them. So we're not like sonicating them or anything. They're simply rehydrating in the water phase and then like pipette to mix for automation purposes. And then we add the dye using the robot for microscopy. So the microscopy is done with Nile red fluorescence. And we are not limiting the size of the vesicles, but there is a limit to what our confocal microscope can see. And so I think the smallest thing we could probably detect is a few microns. I think it would be hard for it to template match anything smaller than that. And obviously the largest things are many, many microns, tens of microns. We don't do extrusion. And I think extrusion is only necessary when you're trying to look at something that's related to size. We don't control for lamellarity at all. Okay. The next question is, could you apply this technique to engineer more stable modern membrane vesicles with phospholipids? Yeah, so that is a really great question. And I think that I would be interested in thinking about different weirder membranes for life, and if they would be more stable. Stable is a hard thing to define in a lot of ways because it could mean many different things. But for example, there's a lot of issues with mRNA vaccines and low temperature stability. And I think that there's an opportunity there to kind of come up with formulations using this method that could improve pharmaceutical applications. The next question is, what about other membrane components? What if you add some peptides to increase membrane stability? Would this be more consistent with some prebiotic co-evolution scenarios? Yeah, I think that we are interested in, so this was a first pass, right? This was our first attempt. I think we are interested in thinking about other molecules that could change the way that our liposomes form. For me, the first thing that I want to do, although we'll see what the group votes on, we're a democratic group here, is I really want to try adding alkanes, because if you're producing hydrocarbons, you're going to have some decanol or decane present, right? Or, some long hydrocarbon chains, some maybe even some like PAHs or other like really what you might think of as like destabilizing oils. And if those are present, can you still form membranes in their presence? For origins of life type applications. I think peptides are hard because, for me, peptides are hard because how do you pick what peptide to use? And a lot of the work in peptides for fatty acid membranes, right? There's some really, Zoe Todd published a pretty comprehensive dipeptide study, and it is not predictive. So which dipeptides you would add are not, there's not like an easy physical chemistry principle underlying which peptides are good and which peptides are bad. And so I think it makes the problem there's too many possible inputs for that. Yeah, but I think, adding organic mixtures is definitely in our future, some type of organic mixture. And the question, this would be consistent with some prebiotic coevolution scenarios. While that is mostly true, it ignores the fact that peptides would be present only if there were a huge number of other organic compounds present, right? Amino acids only occur prebiotically in the presence of a large number of other organic molecules. All right. The next question, could you comment on the stability of these vesicles over time? I cannot, but I would say days, because we make the plates and then we run microscopy not the same day. How long do they maintain size distributions? Do they fuse? Do mixtures of these vesicles with various fatty acids fuse? Kinetics versus time course. So we know that single chain amphiphiles are more dynamic than phospholipids. And some of my previous work shows that fatty acid vesicles fuse over time, and that's a temperature dependent process. They probably also split over time because we're not seeing really huge vesicles only. We see lots of little vesicles still too. We are not trying to control size distribution, although I can see from a cell biology point of view, you might want to do that. It probably, if we are thinking about function, there may be a way to control size distribution by having a fairly impermeable membrane, and then using some kind of like osmotic force to control for making sure that they're like stable over time. But that's definitely a future plan. All right, next question. How do you describe the fatty acids? Is one hot encoding? Have you considered using other physical descriptors? So I think, have you considered using other physical descriptors? Uh, we have. So, for example, what happens if you lump all of the negatively charged molecules in a single pot for understanding principal component analysis, right? And we have thought about doing that. That's part of we obviously have all this data. And so now how many ways can we parse it is one of the questions. And so, yes, that is forthcoming. And our algorithm is not actually thinking about anything about the chemical. It just knows this is the input composition and did it form vesicles or not. And so we're not actually telling it to think about, is this a charged species or a polar species or anything else. I think we have considered using a cost of like how expensive it would be to make the molecule, maybe just by like calculating the thermodynamics of formation or something. And so maybe in the future, we'll calculate based on the cost of formation. All right. In the next comment, there's a comment. I suppose the ML algorithm will need to be able to detect vesicles at all sizes and count them all as positive detection. That's correct. All sizes count as positive detection. Although we could definitely code in their fixed sizes and we know how big they are. So we could very well say, oh, we want to optimize this for a specific size vesicle, and maybe we would be able to get it to be a single size. And there's certainly some compositions that are much more homogeneous than others. So the end part of that comment is about principal component analysis. Could you explore how charge density versus nonpolar head group density impacts the PCA? And I think, yes, that's definitely something that could be done. Okay. I have reached the end of the comments section.
I think you basically gave a second talk answering those questions. That was awesome. Yeah, I'm really glad that someone asked about the peptides, because that would be my especially based on my background with those little prebiotic peptide work we've done, that would be my question, is how to include that. So I was really glad you touched on that. If there are no more questions, I want to say, if anyone does that with modern membranes, sign me up, because I could really use some kind of a high throughput membrane vesicle design prediction stability model.
Yeah, I think if anyone has wants to talk about lab automation too, I think that there's a lot of opportunity right now for lab automation. And so I'd love to just, reach out to me and we can talk about how we set up lab automation on the cheap and what we're doing to optimize it.
Yeah. Especially the on the cheap part, I'm listening. That's been a struggle for us too.
Yeah. Yeah, I think the confocal microscope is not cheap.
Yes.
Yeah, that's the tough part.
All right. If there are no more questions, I want to say thank you so much, Sarah, and thanks everyone else. I see there's another comment. Could you comment on the lab automation setup, including costs?
Really quickly, sure. So we are using an OpenTrons OT2, which is kind of like the standard open source liquid handling robot. It comes with 2 pipetters of different sizes for, when we bought it for 10K, which is pretty reasonable. They now have a new one that has more features. These liquid handling robots can do things like PCR. So the OT2 has like a PCR module on it. It has heat blocks on it. So there's a lot of opportunity for optimizing and customizing it. The challenge is that you kind of need to know Python to make it run. So, with undergrads, that's a tough sell. The motorizing a microscope stage is actually kind of tough. So for our old microscope, they were going to sell us one for like $30,000. And that is not on a confocal. But on the bright side, most confocals do have motorized stages. So if you have one at your institution, you can probably get a 96 well plate option for looking at all of your wells. Ours has a water immersion lens, and it has an automatic water dispenser so that it will run all of the wells without needing to stop to add oil. So that's something that's like kind of custom. And to add that onto the microscope, it was $7,000. But if you don't already have a water immersion lens on your confocal, I have heard that those run north of 50K.
That's not cheap.
Just the lens, right? Just the
Yeah.
microscope lens. So, yeah, I think it depends on what resources you have available. You can always, pay core facility fees for help with that too. So that's our automation setup. We also have a 96 well plate reader. Obviously, we have a fluorescence plate reader, and we're working on assays for those. But I feel like those are pretty standard pieces of equipment now, and so that shouldn't be thought of as included in your cost. Same, these 96 well plates fit in an LC. So if you wanted to do LCMS, you could with a robot.
Yeah.
Yeah.
All right. Thank you. Yeah, I just want to plug a cheap source of plate readers. All plate readers in my lab are from eBay. We never bought a new plate reader, and
Awesome.
I don't think we ever will, because there's perfectly fine ones available. So that's just the one thing that you can get pretty affordable.
Yeah.
All right. If there are no more questions, thank you so much. Thanks, everyone. And thank you, Sarah.
Thanks, Kate.
And
bye, everyone.
bye, everyone. Have a great rest of the week. Bye.